Welcome to a new fMRI tutorial series on AFNI, the Analysis of Functional Neuroimages package. First created by Bob Cox back in the mid-1990s, AFNI has been maintained and developed by a team of scientists at the National Institutes of Health beginning in 2001. Since then, they've added several new features such as linear mixed effects models and surface-based analysis. In this course, you'll learn the basics of AFNI by analyzing a simple fMRI dataset. Then, we'll analyze the same dataset on the cortical surface and examine what advantages we can gain in both statistical power and spatial localization. At this point, you may wonder, why AFNI? Why not one of the other analysis packages, such as FSL or SPM? Ultimately, the decision is up to you, and you'll need to consider many things, such as your operating system and whether your lab prefers one package over another. In any case, AFNI has several unique advantages. One, it's fast. AFNI is able to do many processing steps very quickly and especially effective at quickly estimating models. Two, it's flexible. Each of the major packages offers some degree of flexibility, but in my experience, AFNI's code is the most transparent and customizable. Third, AFNI provides excellent support for its users. The developers are responsive to emails and quick to implement new changes, with new updates being released as needed. The AFNI website also features their world-famous message board, which makes it easy to look up topics or questions. The layout also clearly distinguishes separate threads and clarifies which message is in response to other messages. On the other hand, AFNI does have disadvantages that you should be aware of. First, it can be difficult to learn for newcomers. This has been enough to discourage many people from attempting to learn AFNI, as it requires some fluency with Unix and with programming. Their graphical user interface has also been compared to an airplane cockpit. But if you're willing to face those obstacles, you will be rewarded with an fMRI analysis package that is fast, flexible, and customizable. They also have an odd sense of humor that you may find endearing, or not. So how do you get AFNI? They have a detailed set of instructions on their webpage, and the link is in the text box below. On the other hand, if you're using a Macintosh, you're in luck. You can simply download and run this installer here. Simply double-click on it, and then it will open up. Leaving all the default boxes selected, it will check whether the prerequisite packages are installed, such as Xcode and Homebrew, and then it will install everything. It may ask you for your password in order to install all of the packages. It can take around an hour to install everything, so be patient. To see whether it installed successfully, open a new terminal, type AFNI, and then press return. You should see the graphical user interface open up. Close it, and from the same terminal, we'll type ubersubject.py and then press return. This will indicate whether the Python-based programs are running without errors. And that's it. You're now ready to download a real dataset and analyze it with AFNI, which is what we now turn to. For this course, we'll be analyzing an fMRI dataset that used an experimental design known as the flanker task, which we'll discuss more in the next video. The dataset can be found on the Open Neuro website, an online repository of neuroimaging data. Click on Public Dashboard, and in the search bar, type Flanker. Click on the link that says DS000102. You will see a README summary of the experiment and data parameters, such as the sampling rate and image dimensions. This dataset, like many of the other datasets on the Open Neuro website, has already been analyzed and published. One of our goals in this course is to replicate one of their findings and compare it with what they reported in the paper. To the right, you will see the dataset file tree, which includes the raw data. These include files containing subject data, such as sex and age in the participants.tsv file, and scanning parameters that can be found in the task flanker_bold.json file, which you can either view in the web browser or download to your computer. The imaging data is located in the subject folders, which contain the subdirectories ANAT, which stores the anatomical image, and func, which contains the functional images and onset times for each run. Just like with the behavioral data, you can look at these images individually using the View button, which opens up the image in a Papaya viewer in 3 separate viewing planes. Alternatively, you can download these images individually by using their corresponding Download button. All of these data are in a format called BIDS, Brain Imaging Data Structure. A standardized directory tree, such as this, makes organizing and analyzing the data much easier, as we will see in a later tutorial. Now let's download the entire dataset by clicking on the Download button at the top of the page. The dataset is about 2 gigabytes and comes in a zipped folder. Unzip the file once it has finished downloading. Before we go on to look at the data and analyze it, we should first learn about the task that was used for the experiment, the flanker task. Knowing the details and theory behind it will inform our analysis and what results we expect. Now that you've downloaded the dataset and learned about the flanker task, you'll want to look at your data to inspect it for any problems. As you gain more experience looking at these images, you'll be able to judge whether they are minor flaws that can be fixed by pre-processing the data, which we will discuss later, or whether they are major issues that indicate a problem with the scanner. In either case, you will need to look at the data carefully by using the Afni image viewer. Let's first rename the dataset to something that is clear and informative. If the dataset has been downloaded to your Downloads directory, open a terminal, navigate to the desktop, and type the following: the move command, followed by a tilde, /downloads, / the name of the folder that you downloaded, and then flanker. This will rename the folder to flanker and put it on your desktop. As you saw in the previous tutorial, the dataset has a standardized structure. Each subject folder contains an anatomical directory and a functional directory labeled ANAT and func, respectively, and these in turn contain the anatomical and functional images collected during the experiment. This format is known as BIDS, or Brain Imaging Data Structure, which makes it easy to organize and analyze your data. To look at the data, we will be using the Afni Graphical User Interface, or GUI for short. You can open the GUI by opening a terminal and then typing Afni from the command line and pressing Enter. This will open a controller window and 3 viewing windows. If there are no data in the current directory, a dummy dataset will be loaded. Close the viewer and then navigate to the sub-08 directory within the flanker dataset, and then navigate into the ANAT folder. From here, type Afni and then press Return, which will open the GUI. By default, Afni will look for any images in the current directory, such as files in Nifty or Analyze format, and load all of them into memory. If there are several other images in the current directory, for example, and you wanted to load only the anatomical image into the Afni viewer, you could type Afni followed by the name of the image you want to load. Inspect the image by clicking around in one of the viewing windows. Notice how the other viewing windows and crosshairs change as a result. This is because MRI data is collected as a 3-dimensional image, and moving along one of the dimensions will change the other windows as well. If you have highlighted a window by clicking on it, you can scroll continuously through that viewing plane by using the mouse wheel to scroll up or down. You may have noticed that this image doesn't have a face, which has deliberately been removed in order to ensure the subject's anonymity. If you were to observe that other parts of the brain were missing, however, or if there were distortions in the image, In other words, if you saw artifacts, you should make a note of this. Some artifacts are due to bad data quality or movement or because of scanner malfunctions. Click on the link below for examples of artifacts to look out for. When you are done looking at the anatomical image, you can load in other images while staying in the same directory. Click on the Read button from the menu at the top of your screen. And then in the Directory sidebar, double-click on the file path that ends in 2 dots, which indicates the directory above the current directory. Then, double-click on the Func directory in the Sessions sidebar. This loads all of the images in the Func directory, which you can then browse in the Afni viewer. This new image also looks like a brain, but it's not as clearly defined as the anatomical image. This is because the resolution is lower. It's typical for a study to collect a high-resolution T1 weighted or anatomical image and lower resolution functional images, which are lower resolution in part because they're collected at a very fast rate. One of the trade-offs in imaging research is between spatial resolution and temporal resolution. Images collected at higher temporal resolution will have lower spatial resolution and vice versa. Many of the quality checks for the functional image are the same as with the anatomical image. Watch out for extremely bright or extremely dark spots in the gray or white matter, as well as for image distortions such as abnormal stretching or warping. One place where it is common to see a little bit of distortion is in the orbital frontal part of the brain, just above the eyeballs. There are ways to reduce this distortion, but for now, we will ignore it. Another quality check is to make sure there isn't excessive motion. Functional images are often collected as a time series. That is, multiple volumes are concatenated together into a single data set. You can rapidly flip through all of the volumes, like pages in a book, by clicking on the Graph button and then by pressing the V key. You can stop at any time by pressing the space bar. Note any sudden jerky movements in any of the viewing panes. During preprocessing, we will quantify how much motion there was in order to decide whether to keep or to discard that subject's data. When you're finished looking at the data here, click on the red X button in the top left of the controller window, which will close both the controller window and all of the viewer windows. In the next tutorial, we will learn more about how to pre-process our data to remove artifacts and improve the image quality using Afni's ubersubject.py command.